Right, good morning everyone. Okay, so we're going to start on a series of 2 lectures about antibiotics, and based on feedback from previous years of teaching this is 2 of the favorite lectures of the course, so hopefully you're very excited about this. So we're going to talk about antibiotics and their modes of action, and so I know a lot of you are destined to go to pharmacy school or some sort of clinical, medical-related field, or you have taken antibiotics before in your life. And so it is of interest to you to understand how these things are working against the bacteria in your body. And then we're going to talk about antibiotic resistance mechanisms that bacteria can acquire or just naturally have, and how the bacteria sort of answer back to these antibiotic doses we have been giving them, and how that's created a global problem of antibiotic resistance in pathogenic bacteria. Okay, so the title is antimicrobial chemotherapy, and like I told you in the first lecture, chemotherapy is just treatment with a chemical. So this is completely decoupled from chemotherapy cancer treatments. Okay, so I just want to remind you of that. This is chemotherapy in the context of natural products, which are antibiotics that are being administered. Okay? Oh, yes. Midterm 2, so these are just the statistics from that. So the highest score was perfect score. We had someone get a 250, which is a 100%. The lowest score was, unfortunately, a 43%, and our average is about the same average of every other quarter. It's about a 74.5. So that's just the information about midterm 2. And all the grades are posted, from what I understand. So please do access those. Okay, so now we're going to move on to antibiotics and what they are and how they work, and how, remind you how they were discovered originally. So antibiotics are one of the modern day revolutions in the medical field. Okay, so remember how we talked about Alexander Fleming discovering that inhibitory microbe that was inhibiting the bacteria on his plate. And then later, Florey came and determined that that was actually because of a natural product being produced by that penicillium mold on that plate, which they later called penicillin. So that was in the 1940s that penicillin was really first brought onto the market. That's not that long ago. So many bacterial infections that could literally kill patients have been overcome because of this dawn of the age of antibiotics. And so penicillin was the first one, and now we have a slew of others that we either have been administered to ourselves, or you know about someone else who's been taking these. Okay. So what are antibiotics? So antibiotics are just natural products, most of them. They are things that are produced by other microbes with the goal of killing a neighboring microbe. So microbes are in constant competition with one another, especially in places like the soil, where there's not a lot of nutrients, or there are nutrients in certain areas in the soil, and they're trying to kill one another so they can thrive in that soil. So it's a checks and balances type of situation where you have some fungi or some bacteria that are producing antibiotics, with the goal of just enhancing their ecology. Now what we do as humans is we take that ecology, we study it in the laboratory, and then we purify that ecology in the form of a chemical, which turns into an antibiotic. And so a lot of these are just natural discoveries that happen through antibiotic discovery pipelines. Now, we also can chemically synthesize these once we know what they look like, or we can chemically alter them to make them more potent, or have a longer, more stability, or something like that. Okay? But for the most part, they're just things that microbes make just to enhance their own lifestyle. Okay? So, again, talking about antibiotic discovery. So this is that plate, or a plate from Fleming's laboratory, where he saw this penicillium mold inhibiting growth of bacteria, and that later turned into the discovery of penicillin, which was sort of rediscovered by this natural product chemist, Howard Florey. Later, after Alexander Fleming had noted it, published it, and sort of then like, okay, this happened, this biological phenomenon occurred, but I'm just going to put it over here and not really work on it. until this person picked it up later and said, okay, this is something of use, and after a series of purifications, determined that it was penicillin, and they discovered the structure of that, and now we know what it looks like, and there are now lots and lots of derivatives of penicillin. Okay? This is another image of a little girl who was suffering from a very severe bacterial infection. And this is just showing you how well antibiotics can really work. Okay, so this was day 1 of treatment, all the way up until day 6, and you can see that she's basically completely cured of that horrible bacterial infection. Okay, and so this, again, talking about antibiotic discovery, this is Gerard Demotte. He was a scientist whose daughter actually became very ill with a bacterial infection. And he was experimenting, he was a natural product chemist, experimenting with different antibiotics or potential antibiotics. And he took the very bold move of injecting a drug that was not yet really shown to be efficacious and was still in the discovery phase of that particular drug. But he was so desperate because his daughter was very ill with this bacterial infection, and what he had seen was that on agar plates, where we generally look for competition between microbes or application of a chemical that could kill a microbe, it wasn't really working in that scenario, so on a petri dish. But what he did know is that in his animal trials, it was working. And so he decided, oh, I'm going to do this. I'm going to inject this drug, which is called Prontosil, into my daughter, and I hope that it's going to work. And it did. Okay, and that was the beginning of the discovery of sulfa drugs. So it was a sulfanilamide, okay, which is a sulfa drug, which you've probably heard of. You might have taken it once in your life before. And what it does is it's an analog of para-aminobenzoic acid, or PABA. And PABA is a folic acid precursor. So bacteria can make their own folic acid. We cannot. So we don't have this metabolic pathway in our bodies. That's why we have to eat spinach and drink orange juice or take vitamins or something that is going to provide us with folic acid. So one good thing about one necessary component of an antibiotic is that it triggers or it targets something that is specific to the microbe, but a pathway that we don't have. Okay, so folic acid synthesis is perfect because we don't have that as part of our physiology. So the drug mimics this precursor of folic acid, and it jams the system. And I'll describe how that works in the next couple slides. And if it can't transport folic acid and make it from the precursor, then they can't grow. Okay, and so then they die. Okay, so this is how normal folic acid production works. You have your para-aminobenzoic acid, you have pyridine and glutamic acid. There's an enzyme that will link these and convert them to folic acid. And you can see the simplicity of this diagram, so I'm not asking you to memorize the entire pathway. Just know that para-aminobenzoic acid is a precursor for folic acid, and through an enzymatic process, this is converted into folic acid, and then the bacteria can utilize it and grow. Now, sulfanilamide looks very similar to PABA, okay? But what it does is it gets in here and it sort of jams the system, and it prevents these precursors from linking to one another, and you don't get synthesis of folic acid. So it's like a competition between PABA and the sulfanilamide, and the sulfanilamide wins, and the bacteria can't make folic acid, and then it can't grow. Okay, so I mentioned this, that for antibiotics to be something that we actually want to move forward in the pipeline and actually start administering to patients, they must affect the target organism. Okay, so what is the bacteria that you are trying to kill? Is it a staph infection? Is it Streptococcus pneumoniae? Is it Yersinia pestis or something like that? It's helpful to know the causal agent, okay? And it also needs to not affect humans, right? Or other animals like your cat or your dog that you might be giving antibiotics to, okay? Now, sometimes they can have side effects at very high concentrations, but the doses are low enough to where you don't, these side effects don't manifest. So for example, chloramphenicol interferes with our ribosomes. But if you were to be giving chloramphenicol, it would be at such a dose that it wouldn't reach that level, okay? Some cause allergic responses in humans. So penicillin, right, is, we all know that there's penicillin allergies in the population, and that can be a very big problem if someone is administered penicillin and they're allergic to it. My husband is allergic to penicillin, so I'm very aware of this, and every time he goes to the doctor, I make sure that they know that he's allergic to penicillin. And my college roommate was allergic to sulfa drugs, so there are many different allergies that can manifest in the human population to these natural products. So you just need to make sure that whatever you're giving the patient is going to be effective against the microbe you're targeting, but also not have an adverse effect on them. Okay? In general, these drugs affect microbial physiology, so something that does not exist in humans, okay? So peptidoglycan, we know, is only made by bacteria, so that's the cell wall component. So that's a perfect thing to target, is the cell wall biosynthesis. So you basically blow up the cells because they can't make their peptidoglycan. There are differences in the ribosome structure between prokaryotes and eukaryotes, so you can capitalize on that and target protein synthesis in bacteria. And then you could also target a biochemical pathway that's completely missing in animals, okay? In humans and animals. And so that example would be our folic acid biosynthesis pathway. And you've probably heard of this. So there's different spectrums of activity of antibiotics. You can have broad spectrum antibiotics, and so they can target many different microbes, right? So they have a broad spectrum. And the benefit of this is that they're effective against many species. So sometimes in a clinical setting, you might not know exactly what is causing that ear infection in that child. So you can administer a broad spectrum antibiotic, and it kind of covers your bases in terms of killing a lot of microbes. Now, the dawn of microbiome studies, which we're going to be talking about in the last lecture, has demonstrated that our microbiota are not just passive riders. They are beneficial to us. They help us digest our food. They protect us in invisible armor. They educate our immune system. So now it's this idea of broad spectrum antibiotics might actually be killing some of our good microbes. And so that also needs to be thought about when you're administering these antibiotics. You can have narrow spectrum antibiotics that can be just effective against a few or a single species. And so those are more targeted, and that means we need to know what the infectious agent is if you're choosing one of those antibiotics off the shelf. I mentioned this, that most are discovered as natural products, and so there are constantly lots of researchers and lots of industry, pharmaceutical companies, that go out what we call bioprospecting, and that is looking for natural products out in nature and the microbes that produce them. And can we get them to produce large amounts of these, and can any of this be of benefit to the human race, and also how is it economically feasible to produce large amounts of these natural products? Okay, so some of them can be modified by artificial means, so you can add like an R group or something to increase stability of that particular antibiotic, and some can actually be made synthetically. Okay. All right, there are two types of broad classes of antibiotics. There are antibiotics that are bactericidal, that means they kill the bacteria. And then there are some that are bacteriostatic, and that just means they slow the growth of the organism or prevent growth of the organism. And they're not killing it, but they're just kind of halting the population, which is also effective because that allows the immune system to come in and start doing its job.
On that last slide, you mentioned that it can decrease the toxicity in humans. What's that process? Are you manipulating that? On the first point, like below, some are modified by artificial means.
Oh, okay. So I am not a natural product chemist, so I could only begin to. She's asking, what does this mean? Increased efficacy but decreased toxicity to humans. Okay, so she's asking by what process is that accomplished? It's some sort of organic chemistry that I am not privy to, and it won't need be speculating. So that is something likely proprietary by pharmaceutical companies, or something that a research group has gotten a grant to study and trying to just chemically manipulate a particular antibiotic to make it more effective but not toxic. Okay. So bactericidal, bacteriostatic. So this prevents growth of the organism. It doesn't necessarily kill it, but it allows the immune system to come and clear the infection. Some of these only affect growing cells. Okay, so if it's something that inhibits cell wall biosynthesis, well, cell wall biosynthesis is only happening when the bacteria are growing and dividing. Okay, so penicillin is only effective if the organism is building a new cell wall. And so it has to be growing in order for it to debilitate that pathway. Right? So you have to take all this antibiotic until all the cells have left the stationary phase, and you have slowed the growth of all of them. Okay, so now we're going to talk about MIC and MLC, right? So these are two different ways of calculating potency of a particular antibiotic against a particular microbe. So one of them is the minimal inhibitory concentration, and that's exactly what it sounds like. It's the minimum concentration in which you inhibit the growth of the bacteria. You can do this in an experiment in the laboratory where you set up all these nice tubes full of media that have the same amount of bacteria that you inoculate into there, but they have different amounts of antibiotics. So this is up here. This is the concentration from low to high. And then you can look and you say, all right, here I have cloudy growth, cloudy growth, cloudy growth, cloudy growth. That means it was growing there. And then you start to look where the media is clear, and that obviously the bacteria weren't growing very well. So right at that point is your minimal inhibitory concentration, where you've inhibited the growth of that organism. In this case, it's 1 microgram per mil. Okay? Now, the next layer to that is your minimal lethal concentration. So that is just defined by how much it takes to kill the organisms. So how do you discover that? Okay, so what you would do is you would take aliquots from these tubes and you would plate them out onto media, and then you would see where which tube yields colonies. Okay, so that would be this experiment here. So it's the same experiment, just on a new slide now, where you're taking aliquots of these tubes, plating them on media with no antibiotics, so anything can grow. And you're saying, all right, here, even though I inhibited growth, there were still bacteria in there that were alive, and I can see that. And I can see that here, and I can see that here. And so this is the plate, the concentration that gave you a plate with no bacteria growing. So that's your MLC, or your minimal lethal concentration. So your minimal lethal concentration is always higher than your minimal inhibitory concentration. So for this example, that would be 4 micrograms per mil was our MLC, and our MIC was 1 microgram per mil. Okay, so like doing something like that looks like it takes a long time, right? Like you need a lot of tubes, and if you're a good scientist, you will be replicating that, not just doing it one time. You would have to do it several times. So that's a lot of tubes, a lot of media, a lot of waiting, a lot of incubator space. So there are quick and dirty tests that you can do to look at this. And one of them is called the E-test, and this determines the minimal inhibitory concentration, the point at which, or the concentration at which you inhibit the growth of the microorganism. And this is where you have a strip that is impregnated with increasing concentrations of the antibiotic. So you plate a lawn of your bacteria on your petri dish, and you lay this strip over it. The antibiotic is going to diffuse out of the strip into the media, and then you're going to look for growth. So here you see that it's growing, this cloudy area, and it indicates growth. And then you look right here where it starts to inhibit growth, and that is 0.047 microgram per mil. You just have to trust me on that. That is what it says. And that is your MIC for that particular microbe against that particular antibiotic. Okay? All right? There is another more, elegant experiment and more robust experiment called the Kirby-Bauer DIS susceptibility test. So this tests one antibiotic. You can only put one antibiotic on there. This tests many antibiotics at the same time, right? So if you are in a clinical setting and you're trying to look at this isolate that's come from a patient, you're trying to decide what antibiotics can I give this person, it's nice if you can get a lot of information at once, okay? Because this can take a while. And you don't want to be sitting around for a week waiting for results and like, okay, and then the infection has progressed. So you can look at this using a lot of different discs at the same time. So this is the Kirby-Bauer test, and it tests multiple antibiotics. So all these discs, so again, you start with plating a lawn of bacteria on your Petri dish, and then you come back and you put all these discs. So each disc contains a different antibiotic. And then those antibiotics are going to diffuse out into the media, just like it did up here, but it'll be a circle instead, because it's a circular thing. And then you'll look at the zone of inhibition size, for example. And so if it has a very small zone of inhibition or no zone of inhibition, that means that that microbe is resistant to that antibiotic, because that antibiotic did not impede its growth at all, right? So oxalisin, in this case, this microbe is resistant to that. Now, if you look at this zone, it's pretty large, and that is a sulfa drug called trimethoprim, and this bacteria is very susceptible to that. It's very sensitive to the sulfa drug, because you can see that that sulfa drug is preventing its growth. Right. So this is, again, how the Kirby-Bauer assay works. You inoculate your bacteria here, and then you are going to place your discs onto the plate. And there are even machines that will just put them all at the same time, so you don't even have to do them one by one. And then you incubate and let them grow, and then you measure the zones of inhibition. Okay? So not only this is important for a clinical setting, but sometimes for epidemiology. So if there is an outbreak of an antibiotic resistance microbe, many different clinical labs across the U.S. or across the world can do these tests on their isolate from their hospital or wherever, and then they can calculate the sensitivity or resistances to specific antibiotics, and then they can compare and contrast. Okay, say, all right, I think we might have the same isolate because we're getting the same amount of resistance to this class of antibiotics from isolate in Michigan versus isolate in California, for example. Okay, so anytime you're trying to compare data from one lab to another, you have to have a high amount of standardization. Okay. All right, so I'm just going to skip to the next slide to talk about that. So you need to make sure that all of the labs are doing the experiments the exact same way. So this means that you're going to use the same agar plate, because this is all based on diffusion rates of chemicals. If you have a bigger plate with a thinner amount of agar, you're going to get quicker and faster diffusion than if you have a thicker plate. Okay, so you need to use the same size plate with the same amount of media. So you have to actually measure this very carefully. It's not just like pouring plates until they look sort of fullish. You have to provide the same depth of the media by pouring in the same amount. You have to use the same media. So this is Mueller-Hinton agar. And then you have to put the exact same number of organisms to spread on the agar plate. So whatever you're inoculating the plate with, your concentration of bacteria needs to be the same. And the size of your disc and thickness of the disc needs to be the same, because that will also have an effect on the diffusion of the chemicals outside of the disc. Okay? And then you need to use the same concentration of antibiotic in the disc. And then you need to incubate at the same temperature. And from that, you can then draw conclusions that you can compare with your sister lab that might be in North Carolina or somewhere like that. Okay? All right, from that, you can get information, like I said, about the minimal inhibitory concentration of a particular antibiotic against a particular microbe. And this is useful because when you're administering this to a patient, the MIC has to be higher, the concentration in the tissue that you're targeting has to be higher than the MIC or it's not going to work, right? That's logic. So you need to make sure of that to ensure the effectiveness of the antibiotic, right? Now, that can be problematic because if you give someone a high dose of antibiotics, that can lean towards side effects, and you don't want that. So how do we make sure that the MIC stays constant and is related to the dose of the antibiotic? So that's why you take repeated doses, right? So when you get your antibiotic, your amoxicillin, it says take it 3 times a day, and your doctor tells you make sure that you don't really change those times a day. That's because they're trying to keep the minimal inhibitory concentration, the dose that's in the tissue higher than the minimal inhibitory concentration, okay? Okay, So now we're going to talk about fun stuff, which is the mechanisms of antibiotic action. And we already talked about how it has to be something that bacteria have that we don't have. And so these are the major targets we're going to talk about. We're going to talk about cell wall biosynthesis, cell membranes that are different in prokaryotes such as eukaryotes, DNA and RNA synthesis, protein synthesis, and metabolism. Okay, so here is a list of antibiotics. And every quarter, students come, do I need to memorize the list? The answer is, if I say the word in class, yes, you need to memorize it. If I don't mention it, then probably not, right? So penicillin, I've already said that like a lot of times. So you need to know how penicillin is going to work, which I'm going to explain to you in just a second. We're going to talk about bacitracin, vancomycin, tetracycline, and aminoglycosides, polymyxin, and also the sulfonamides that we talked about earlier. And we're going to talk about rifampin here. Okay. All right. So this is lifted from another textbook, but I liked it because it was kind of summarizes everything that I'm going to talk about in one slide. So these are all the different things we can target in terms of antibiotic effectiveness. And so we're going to talk about cell wall synthesis. So this is our peptidoglycan. We already talked about folic acid metabolism. DNA gyrase. So the DNA gyrase of prokaryotes is slightly different than eukaryotes, and that can be a target of antibiotic action. And we're going to talk about protein synthesis, 50S and 30S inhibitors, and also disrupting the cytoplasmic membrane. So that's basically just disrupting the membrane in such a way that you cause it pores to form, and then the bacteria will lyse because of that. Okay, so peptidoglycan, just to remind you, we're bringing back some concepts from the first set of lectures. So remember that it is sugar and connected by these peptide linkages, and the sugars are N-acetylmuramic acid and N-acetylglutamic acid, and that's NAG and NAM here. And all of that is then polymerized, so you'll have these repeats of NAM and NAG connected to another NAM and NAG, and so on and so forth, and down here, and that is connected by a peptide bridge, okay? They're connected by peptides either through an interbridge or direct, connection between these peptide linkages, okay? So that's gram-negative or gram-positive, but the same sort of process takes place in both, okay? And you can have antibiotics that target specific steps of this, right? So the one we're going to talk about, or sort of a class we're going to talk about, are the beta-lactams. And penicillin is a beta-lactam. So that just means it has a beta-lactam ring, and there are lots of different versions of beta-lactam rings, and they just differ by their R group, okay? So an example is penicillin. We also have amoxicillin, which I'm sure you've probably been given, especially as a child for an ear infection, ampicillin and carbenicillin. And the beta-lactam ring, which is this part here, is the key feature of these compounds, okay? And they target cell wall biosynthesis. We also have vancomycin, okay? And so that targets just a slightly different step of cell wall biosynthesis. It's very similar to that mode of action of penicillin, but also different. So it's a different antibiotic. Cycloserine can block formation of the peptide for the cross-link, and bacitracin blocks movement across the membrane. So I'm going to show you that in a diagram here. So this is just the general steps of how we make peptidoglycan. Okay, so the NAM and the NAG are cross-linked in the cytoplasm. So this is the cytoplasm, and this is the membrane. And this would be outside of that membrane where the peptidoglycan will then be building the cell wall. Okay? So what first happens is you have these peptides being linked to the N-acetylmuramic acid in the cytoplasm. Cycloserine inhibits that step of the dealanine to the dealanine. Okay, so if you block it there, then you're not going to get the rest of this happening. Okay? If it were going as normal, then the N-acetylmuramic acid with all of its peptides attached to it would then link to the N-acetyl, the NAG here. And so then you have the subunit of the cell wall. And that is linked to a polyphenol phosphate carrier in the membrane, which then flips all of that to the other side of the membrane where it needs to be built. So the subunits are built in singlet, and then they're flipped, and then they are linked together out here on the other side of the membrane by transglycosylases or transpeptidases. Okay? So transpeptidase is linking the peptides together, and a transglycosylase are linking the sugars together. Okay? So glyco and pepto, you can derive the meanings from that. So we have drugs that will inhibit the transpeptidases and the transglycosylases. This is our penicillin and our vancomycin drugs here. So if you inhibit this part, then you're going to stop all of this, and you won't have any linked peptidoglycan. Okay? Now, in bacitracin, this affects the flipping of this over the polyphenol phosphate carrier, flipping back into the cytoplasm, and also linking that carrier to this N-acetylmuramic acid peptide linkage here. So this is what this prevents here. And sort of a fun fact is that bacitracin, you cannot take that internally. It's bad for your kidneys, but you'll find it in topical ointments like Neosporin and other things that you can put on your skin. So next time you have Neosporin, you can look at the ingredients. Say, oh, I remember that from 121. And then if you're doing Trivial Pursuit or something, it might come into play. I don't know. Who knows? You look really smart. Okay, so these are our beta-lactams, like I was talking about. So this is our basic structure. This is our ring. These are our R groups, and so you can have different R groups. I'm not going to ask you to know what those R groups are, just to know that these drugs all share the same similar structure of a beta-lactam ring, and they differ by their R group. Okay? Now, these are very susceptible to degradation by beta-lactamases, right? So these are enzymes that are produced by microbes to degrade this. So this is how a microbe will answer back. Okay, I'm in this environment where I have a lot of this beta-lactams that are trying to, drag me down by inhibiting my peptidoglycan biosynthesis, and I can make this enzyme and I can just degrade that. And they are very potent. Okay, so even at small concentrations, you can get these beta-lactamases that are very efficient at degrading this. Okay, and this can actually be a pain when you are trying to grow bacteria in the laboratory that you are resistant to these antibiotics, and you need to keep supplying the antibiotic because they're so good at degrading it. Okay. What else was I going to say about beta-lactamases? Okay, so other modes of antibiotic action. So we talked about nalidixic acid, so that targets DNA gyrase during DNA replication. So that's the enzyme that unwinds the DNA, so that DNA polymerase can come and start replication. And that's a quinolone. Okay, so that's our nalidixic acid. We already talked about sulfa drugs. Okay, so this inhibits actually a metabolic pathway. So if you're going to be splitting hairs and you're reading the literature and you start to work in this area, it's actually really defined as an anti-metabolite because it's preventing the production of a metabolite. Okay? And that metabolite is folic acid. So you can see here that para-aminobenzoic acid, which is our precursor to folic acid, looks very similar to sulfanilamide. And so you can see how this could, if this is in the system and it outcompetes this, it could really jam that system and prevent synthesis of folic acid or vitamin B9. Okay, so I'm talking really fast. I only have a few more slides. Okay, so this is our RNA synthesis inhibitor, which is rifampicin or actinomycin. So RIF is a very interesting drug in that it's red. So if you're ever using this in the lab, you're like, oh wow, they have like this really red solution here. It can also turn urine and breast milk sort of an orange color. So patients need to be aware of that if they're given this drug. I don't think it's commonly prescribed. I'm obviously not a clinician, but I've never been given rifampicin, but maybe in some cases that they might need to utilize that. And what it does is it inhibits transcription, okay? And rifampicin, or RIF for short, blocks bacterial RNA polymerase, but it doesn't have an effect on human RNA polymerase. And that obviously it will inhibit the transcription process if your RNA polymerase is not working very well. Actinomycin D also inhibits transcription, but what it does is it binds DNA and inhibits the polymerase from moving. Okay, so the RNA polymerase has to move down the DNA to transcribe that into messenger RNA. And if actinomycin D is there, it's binding to the DNA and preventing that process from happening. And a very common set of antibiotics are ones that inhibit prokaryotic ribosomes. Okay, so we're going to talk about several different ways. Some of them inhibit different processes of the ribosomal protein synthesis mechanism. So macrolides, like our chloramphenicol that we've talked about, they bind the 50S subunit, or the large subunit, and what they do is they block the transfer of the peptides during protein synthesis. And so this will halt protein synthesis here. And our examples of that can be our chloramphenicol or our erythromycin or azithromycin. Okay. Aminoglycosides, they prevent the 30S and 50S subunits from binding to each other. So remember the initiation of translation. Once the messenger RNA is locked into the 30S, then the 50S will dock down on it, and then translation can occur. These drugs prevent that from happening. And our example of that is streptomycin, which is produced by streptomyces that have been isolated from the soil. So I mentioned that the soil is a great place to find competitive interactions. Well, streptomycin is one of those, and it's utilized as an antibiotic in humans and also in agriculture. And so it's registered to be sprayed on specific crops to kill bacteria that infect plants as well. Tetracyclines here, they bind the small subunits, the 30S subunit here, and they block binding of the aminoacyl tRNA. So they prevent these from coming in here, and that will halt protein synthesis. Okay, and then our last piece is talking about the ones that disrupt the cytoplasmic membrane or the inner membrane or the plasma membrane of the bacteria. Okay, so what they do is they're very sneaky and they can get incorporated into the membrane because they have similar sort of structures to them. But they form pores, okay, and they damage the integrity of the membrane such that it lyses, okay? So polymyxin is one I'm going to pick out here as our example, and you can also find that in your tube of Neosporin. There are three antibiotics in there, and polymyxin is one of those, and it disrupts the lipopolysaccharide layer and will lyse bacteria that way. Okay, and then gramicidin is our other example of ones that will form these channels with these pores. Okay, so this is the gramicidin channel. It basically forms a cation channel, and you get a lot of hydrogen ions leaking out. And remember how maintaining that balance of those protons is very important for maintaining the proton motive force. And if you disrupt the bacteria's ability to maintain the proton motive force, it can't make ATP, it can't do a lot of other processes, and it will die. Okay? And again, it's usually only applied topically because it can also, if you ingest it, can depolarize human cell membranes, and that's obviously not a good situation either. That's all I have for today, and I just want to remind you, because I got an email this morning, that IEVAL is operational. And